also a VCP patient or a mutation, a patient with a VCP mutation. Pleased to introduce our speakers for session 5, which is entitled Drug Discovery, New Inhibitors, Small Molecules. This is going to be a great session. This may be a provocative session, and that's the word that when we were talking to Donna and Tsui-Fen, that we knew that there's lots of opinions about inhibitors, and even after this session, we're going to have some of the abstracts that addressed inhibitors, and we love the Q&A. Again, feel free at the Q&A or the question and answer time at the end to, turn on your mic and camera and ask those questions. Let's have a great discussion. So let me introduce our speakers. So we have Dr. Donna Huryn, professor at University of Pittsburgh School of Pharmacy. We connected a couple years ago with a great journal article that she had kind of summarizing inhibitors. And then we have Dr. and Tsui-Fen Chou. Tsui-Fen, I know I probably messed up your name again, but research professor in biology at Caltech. And, I want to recognize Tsui-Fen because this conference, she really has pushed us and been helpful and offered to host if we're going to do it at Caltech. So, Tsui-Fen, I appreciate your persistence and encouragement on that. So they're going to lead a discussion on recent drug discovery findings and mechanistic studies of VCP inhibitors, including small molecule compounds and ASOs. And so I want to welcome Dr. Huryn and Dr. Chou, and I am going to shut up. So Donna, if you want to take.
Yep. Great. Thank you, Nathan. And thank you, Sanjita. This has been a really exciting day yesterday to hear so many people so passionate about VCP and the science behind it. So I think Tsui-Fen and I are going to talk a little bit about inhibitors, and I'll take you through some of the, just a general overview, and then some of the work that we've been doing in collaboration with Tsui-Fen as well as Michelle Arkin. So the good news is that over the last maybe 7 to 10 years, there's been a significant increase in people talking about inhibitors of VCP. And so the hope is that this only continues, and that by having better inhibitors, more well-characterized inhibitors, that we'll be able to use these really to understand p97, what it does, and actually start putting even more compounds into clinical trials to see its effects. But really before 2010, there were kind of modest tools as inhibitors to use. There were genetic methods, people used siRNA. There was some small molecules reported, particularly this natural product called iarastatin, which was identified because of its effect on ERAT, and they only figured out later that it bound to p97. Despite that, its mechanism is not well understood. There's still a lot of studies done on it. It appears to be accumulate in the endoplasmic reticulum. That's part of its effects. But I think, and I'll start being provocative. Natural products sometimes they're great, but sometimes they're also challenging because they often have many effects that are unknown, or they're also metabolized. There's other natural products called withaferin. There are reports about gossypol, which has huge numbers of polypharmacology, and so when you do these, say, oh yeah, it inhibits p97, but it does many other things too. So it's really hard to interpret some of the data with some of these small molecules. But really, around 2010, 2011, we started seeing the first directed programs to look for inhibitors of p97, and the first ones, important ones, came from projects that Tsui-Fen was involved in through the molecular libraries screening centers that NIH had set up. This was through Scripps. The chemistry was done at Kansas. Ray Deshays contributed to that as well. And these were particularly these aminoquinolines, particularly DBAC, the ML series, which was really the inspiration for some even more important compounds. Those programs are really directed towards probe molecules, and so they had some level of potency, perhaps cell-based activity, but the goal wasn't to make drug-like molecules to do further, and so they were limited by just their characterization. And at that time, there was really no structural information about how they bound, although it was known that they were active site inhibitors. So in the past 10 years, though, we now have really more of an explosion of people working in the area. You see new people publishing that hadn't published before. There's just much more interest. There's many different scaffolds in addition to the active site inhibitors. We now have allosteric inhibitors that our groups have worked on, and I'll talk a lot about those. And because we just have more knowledge of it, the compounds we know are a little more selective. They're certainly more drug-like. We now have compounds that are active in vivo. And they're more thoroughly characterized. And so what that allows us to do is really to study VCP and its role in disease with these inhibitors. And so they're really tools that really can help understand the biology and as starting points for drug discovery. So just really briefly, I think Tsui-Fen's going to talk a little bit about this, but an important series were these quinazolines that started from DBAC and high throughput screening. You could see in the blue is where they made the structural modifications to make these more potent. They advanced to this compound in the middle, and then that evolved to some really important compounds. The one on the left is a covalent inhibitor, p97. These are all active site inhibitors. You'll see a lot in the literature about some of these compounds. And then the two compounds that have entered clinical trials is CB-5083. That went into phase 1 for cancer. There were some off-target effects that limited its ability to proceed further, so those stopped. But there was other compounds. This was supported by and came from Cleve Bioscience that eliminated that off-target effect, and now this compound 5339 is in clinical trials for cancer. And hopefully we'll start to see those results probably within the next year. We now have X-ray structures of CB-5083 or cryo-EM structures bound to p97 and VCP. It binds again at the D2 site, the D2 ATP site. It's a competitive inhibitor. And what's on the right is the yellow is CB-5083. The red is an analog of the nucleotide that's bound to the active site and just shows that this is a competitive inhibitor. They bind to the same site. So the work that I'm really going to focus on today was under the auspices of this NCI Chemical Biology Consortium, which started about 10 years ago. And the focus was to really put compounds into phase 1 clinical trials for cancer, particularly for projects where there were challenging targets, there were risky targets, maybe it hadn't been validated, and for which there was an unmet medical need. And so they had proposals come in, and one of them was through for p97. So when it started, it was a much smaller group of people. We were at the University of Pittsburgh, where I'm the PI. Michelle Arkin, who'll be talking later, this group at UCSF was involved. And then about 5 years ago, they significantly expanded it to include 23 centers. And so we have centers who do a lot of different work, who are these dedicated centers who work on multiple projects, contribute multiple things. And then we have very specialized centers that do very specific specialized activities, for example, PK studies, right, or cryo-EM studies, for example, that come onto the project when it's needed, and then maybe exit the project when that work is completed. And so that's what happened in this project, and it started really early on when Ray Deshays wrote a proposal with the University of Minnesota to bring p97 and VCP as a project into the consortium. And early on, UCSF, University of Minnesota, and SRI International started to develop tools that we needed to do this project: assays, screening platforms, cell-based assays. They ran high throughput screens, and then when they started getting hit molecules to work on, we became involved to do the medicinal chemistry. Tsui-Fen became involved to contribute her specialization in p97 and cell-based assays. And then we also got access to resources at NCI, the cryo-EM lab, for example, the biomarker lab. Most of the work I'm going to talk about today was early in the project, but more recently, things again has evolved. University of Minnesota, SRI International have come off the project, but we have the addition of some important partners, particularly CROs, Pharmaron, AMRI, and Extal. And so when the group at UCSF ran this high throughput screen, one of the molecules that came out was this phenylindole that appeared to be really promising. It had modest activity in the isolated enzyme assay. They could tell it bound to full-length p97 through an uncompetitive mechanism. And so that was really intriguing because it differentiated it from the quinazolines and the CB-5083-like compounds. Importantly, it had really good properties. It was drug-like, it had good solubility, and so we could do a lot of things with these molecules. We just needed them to be more potent so that we could really start to pursue more cell-based activity, understand it, and even some in vivo activity. So I don't do this justice, but we spent years making molecules around this to optimize the structure. At the time, we didn't have structural information about where exactly it bound and how to get better compounds. So we had to do a more empirical approach where we modified virtually every portion of this molecule to find better compounds. And these were not trivial syntheses. They were 8-10 steps. They required a lot of development. And I just want to highlight that it's, you see inhibitors in the literature, but it's actually a lot of work behind that. But while we didn't have structural information, the information we got when we did this structural activity relationship study really gave us a lot of information about what the corresponding binding site on p97 was going to look like. And so, excuse me, so what that told us was we knew we needed the specific conformation in the orange. And so it showed us that the molecule and the binding site was probably curved. We knew that this section on the left with the purple and green was highly sensitive to any changes. And so that suggested it was deeply embedded into the protein. We knew we needed specific hydrogen bond donors and acceptors in this blue region. And that suggested there was complementary donors and acceptor on the protein. And then we also knew that we needed some lipophilicity on this far end. Again, so the complementary region on the protein would be a lipophilic group. And then toward the very end of this, we actually got a collaborator at, excuse me, at NCI, the Subramanian lab, interested, and he solved a high-resolution cryo-EM structure of not only p97, but p97 bound to our inhibitors. And that the classic view from the top is shown here. Our inhibitor is red here, and as we expected, we knew it bound to the D2 domain. But now we could see exactly how it binds. And so it sits here very close to the D1 interface. And you can see the structure is in this down conformation that we've been talking about. And so when we talk about p97, and I think this is something that the group has, we think of it as sort of a set of gears, right? And so we talked a little bit about this way. There's this ATPase activity in that D2 domain in the blue that drives the motion. And that gets transferred into effects in the D1 domain and eventually in that N domain. And basically what we've done is really shoved a wrench into those gears, and we've locked it into an inactive conformation where it's in that down conformation. The close-up of the binding site really confirmed our SAR studies. You can see here it's kind of curved. That indole is deeply embedded into the protein, and then we have a little more flexibility, at this other end. We could optimize these even further by incorporating some medicinal chemistry strategies, and now we have compounds that are sort of 20 nanomolar, and we think we understand why they're even a little more potent based on some docking studies based on that original cryo-EM structure. So with these tools now, we can actually start doing a lot of things. And so one of the things that we did, which was kind of interesting, is we used it as a tool to perhaps think about new methods to enable structural information. And so we conjugated these small molecules to these lipids and then started immobilizing them on these affinity grids for cryo-EM. And so when we did that, we can then pull down, it's acted as a bait, it pulls down p97, and then you can get high concentration of p97 on these grids, and then you can solve the structure. And so one of the structures solved is this unusual dimer that we saw yesterday as well as one of the structures that we solved when we did this. And so this is just a tool, but what it offers is the opportunity to do things like, what does p97 look like in a cell? If we could use this as bait, we can pull out p97, see what cofactors are bound to it when it's bound to this inhibitor. So this was really just a proof of concept study, but it allows us, I think, to start thinking about other ways to think about what these molecules are doing, these inhibitors are doing in the context of a cell. So a second series that we've been working on is based on a report that was in the literature from Nerviano and in collaboration with Genentech on these triazoles. It started again with a high throughput screen. They published this compound, this tool compound, NMS-873, which we see in a lot of papers. It also works through an allosteric mechanism, but it's a little different from the phenyl-indole. So these are non-competitive, and so the mechanism is slightly different. We have worked on these a lot. I won't go into a lot of detail, but we further optimized them. Here's a couple of examples where we modified the blue region, we've modified areas in the red region. We have much more potent compounds now. We have compounds where we see in vivo efficacy through this allosteric mechanism. And just again, we have access to the biomarker lab at NCI, and they've been able to show that these compounds, these are the allosteric inhibitors, these UPCDC numbers in cells do the same thing as the active site inhibitors. So they cause accumulation of ubiquitinated substrates, they cause accumulation of CHOP, an increase in CHOP, and then included caspase-2. And so it looks like the allosteric inhibitors can have the same effects as the active site inhibitors. And these compounds are potent enough to do that. Recently, there's been an X-ray structure of NMS-873 bound to p97 reported, and that's shown here. Again, it gives us a lot of information about what to do. It's an overlapping binding site with the phenyl-indoles. It's slid out a little bit. It's not, there's a little, some differences that might be important to the different mechanisms. But what we now have here, and this work is, that I'm going to talk about next, is really done through Michelle and Tsui-Fen, is we have these three molecules that work three different ways. And so we have these allosteric inhibitors in red, these phenyl-indoles that work through an uncompetitive mechanism. We have the CB-5083 series, these quinazolines that are competitive inhibitors, and shown here in the teal is the nucleotide, so they bind here. And then we have these triazoles that are non-competitive inhibitors. And so we can start asking, what are these all the same, or do they different? And we can start interrogating VCP. And so just in general, here's what these structures are. They're all very potent in an isolated enzyme assay. Most of them have good properties. As I mentioned, these triazoles don't have great properties, but we have better compounds than these now. And so what we can start to do is, you saw a slide very similar to this, or almost identical to this yesterday, is start asking, what do these do in cellular assays that look at each of these different pathways that p97 is involved in? So when you look at just a general anti-proliferation assay, cell titer glow, you see the compounds have virtually very similar effects, right? And so potent activity at ATP is about the same types of potency in proliferation. But we start to see differences when we look at very specific pathways. And one of the first differences we saw was the ability to stabilize a UBGFP. So this is a measure of the reflection of the ERAD pathway, where CB-5083 is very potent, but this uncompetitive inhibitor is not. The non-competitive inhibitor does see effects. When we look at autophagy, we see something very different. And yesterday we also heard about autophagy, and in that case, the uncompetitive inhibitor is effective, but the non-competitive inhibitor is not. And then in a third study, when we're looking at the role of p97 in Golgi reassembly, we see that the uncompetitive inhibitor has the most profound effect on inhibiting this, while the competitive inhibitor has some effect, and the non-competitive inhibitor does not. And so what we think we're seeing is context-dependent effects, right? So now we have compounds that work in these two pathways, another compound that only works in the ERAD pathways, and then a third compound that works in two different pathways. So we really need to investigate this further. I think our current one hypothesis is that these molecules are affecting p97 in the context of one of its cofactors very differently. And the assays we use are just isolated p97, but p97 doesn't act in isolation. And so it probably has to do with how these molecules, inhibitors, inhibit p97 cofactors that we still have to study. And so just in closing, sort of where we are as of last year is that really most of the work for the inhibitors has gone into focusing on cancer, right? And so it's in some ways it's a little easier. You're looking at wild type, you're looking at the therapeutic approach is inhibition, and it's relatively straightforward what you want to do, unlike VCP disease, right? And we have small molecules that there are many examples with in vitro activity, there's in vivo activity reported, and we have the compounds in clinical trials. So these certainly are the most advanced. More recently, we've seen effects of reports of the effect of p97 inhibitors as antivirals. And so many different influenza, polio, HMCV, for example, and even COVID-19. And so we see a lot of this, and so that it will be interesting to see whether this becomes a therapeutic approach to antiviral therapy. Much less available are effects of inhibitors in models, of neurodegenerative diseases, for example. There's some of the ones shown here. So there are some preliminary data for quinazolines and the triazoles for these probe molecules. There's certainly data in Drosophila models for some of these, but that's certainly an area where a lot more could be done, especially with these much better compounds now. It also is much more complicated because, as we'll probably talk about, we don't know whether we want inhibition or activation. And there's also the question of mutant p97 versus wild type. Other kind of interesting things to think about, or we think about, is that there are recent reports of the role of VCP in cardiovascular disease, and is it protective? And so that raises the question is, if we have inhibitors, is this going to cause problems, cardiovascular problems in patients? Some of the studies that were done, particularly with COVID-19 and these inhibitors, suggested that we may not see a great enough separation between effects on antiviral effects and just general toxicity. And so that remains to be seen. And then there's also intriguing data in Huntington's disease, but they used a non-selective natural product. And so it's unclear whether how to interpret those studies. But I think the really cool thing is that there's amazing things that are really right on the horizon for VCP. As we saw yesterday, just beautiful structural studies. We have now inhibitors of various mechanisms. We're starting to understand why these inhibitors work. We have VCP cofactor structures, and we have VCP cofactor inhibitor structures. And the hope is that we will get more of these. And it raises the possibility is that we can design specific inhibitors that target VCP and a specific cofactor and not other cofactors. And there's certainly evidence in the literature to suggest this is possible. We haven't, they've been more retrospective, but it's certainly something that we think about. And is this something that we can do? The structural information will have that. Another big question is, can we make inhibitors of the mutants that don't inhibit wild type? And so again, that's something that maybe the structural information will help us with, and that's probably a tall order, but something that we can certainly strive to. But I think with the availability of these small molecules now, multiple molecules working in different mechanisms that have very good properties and are likely selective, we now have an opportunity to test them in multiple disease models and see what the role of VCP is in all these different diseases. And finally, the holy grail, at least in my field, is can we make activators from inhibitors? And we'd love to think that we could. And I think our hope is that if we found molecules that lock VCP in an inactive conformation, why can't we make molecules that lock it in an active conformation? And so I think that's certainly something that would be exciting to do, and certainly something that we think about. So just in closing, I want to thank the work is a huge effort by many different people. I particularly want to thank my collaborator, Peter Whiff at Pittsburgh, Michelle Arkin and Tsui-Fen, and also highlight the cryo-EM work was done in the Subramanian lab. David Thompson at Purdue did the work on the p97 inhibitors as bait. And then also need to thank Ray Deshays for his contributions, and those at Leidos and NCI for their support of this. It's really exciting to work on such an important project, one that's so exciting and sort of just at the brink of sort of getting drugs for really challenging diseases. But it's also really nice to work with great people and have fun doing it. And so here's just a picture of us at a consortium meeting. And here's Michelle Arkin and Peter Whiff. There's one of our posters on our inhibitors. And I'm pretty sure Tsui-Fen is like right over here with her son. This was Tsui-Fen at the meeting at UCSF. And so I just wanted to thank all them for making it such a great project to work on, to have such great collaborators.
All right. Thank you, Donna. Tsui-Fen, you want to go ahead and share your screen?
We'll try.
I got it for you, Donna. We did have a question from Chris. I think Peter's question was answered, but while Tsui-Fen's getting set up, do you know if the inhibitors that affect different VCP functions is different in different cell types, tissues?
I don't know that we know that, but maybe Tsui-Fen will answer that. I know she's been looking at cofactor, p97 cofactor, as far as inhibitors, but I don't know if she's looked at different cell types.
All right, great. Well, we'll keep that in mind, especially towards the end. All right, Tsui-Fen.
So can you see my slide?
We can, yep.
Okay, great. Okay, so thank you, Donna, for this nice talk. So a lot of information will be very useful when I discuss our project. And also I'd like to thank Nathan. When my lab first moved to Caltech in 2019, the first day, Nathan and Allison show up to the lab and introduced to people in the lab, I think inspires us to work harder. And so my title is Chemical Proteomics Profiling of Several p97 Inhibitors. So this slide is not necessary, but the only thing I want to point out is the first substrate link p97 in East called CTC48, or we call VCP, is the discovery at Alice Vasharsky's lab. He used yeast system to discover this ubiquitin fusion degradation pathway. And this is the artificial substrate. This is not happening in nature, the one he used. But this shows us how important this genetic model organism tool is important for us to even understand the protein function. So his lab shows p97 in his CTC48, and a few UFD gene is important for degradation of this reporter substrate. So since then, there are many functions has been discovered in different labs. And when I first started as a postdoc in Ray's lab, I actually start with the e-screen project, try to find the substrate for different cofactor for p97. As my side project is to try to figure out if there is a way to find a p97 inhibitor, since my training as a PhD is in medicinal chemistry. And at the time in the field, the validated drug target is the proteasome. So if you look at the timeline I put on the right, 1994 to the first drug being approved by FDA, it's less than 9 years. And the important thing is this pathway is a good target, but there are some off-target effect or cytotoxicity side effect that we think might be able to overcome by blocking the upstream of the UPS pathway. And when I start at that time, the literature we know p97 does not function by itself. And this is a nice review already in 2009. You can see a lot of cofactor, a majority of cofactor bind to the N-terminus domain, and a few of them bind to the C-terminus. And on top of that, p97 is also phosphorylated, and the function of that is still need to be studied more. So why do we need a specific p97 inhibitor? One thing we know is, well, with a good pharmacological inhibitor, we can use as a research tool to study p97 biology. Because at the time in the lab, if you want to know if a substrate is a proteasome, if it's a protein is a proteasome substrate or not, we can simply purchase a compound called Mg132. And you do the experiment. If you see the protein degradation slow down by Mg132, indication this substrate is a proteasome substrate. By the time, there is no good p97 inhibitor. And second, it's a good opportunity to develop drug to target p97. And the way I start this project, which is interesting because the first day I come to the lab and we discuss, and Ray connect me to Brian Stowe's postdoc. So we just start to talk about how to design inhibitor. And I spent a month to look through literature and try to find all of the structure I can to look at it. And we first designed this covalent inhibitor that targets cysteine 522. And the compound Donna explained derived from CB-5083, those all target the same cysteine, this cysteine 522 in this structure on the right. So we finally published this paper this year to summarize this work. And so Amanda quickly make a few compounds, and one of the compound I always call ACZ99C, now in the paper we call PPA. So you can see this compound has this microacceptor can react with the cysteine 522 in the active site of p97. And this is the in vitro assay to look at purified p97, purified NSF, and purified yeast for CTC48, and shows it only inhibit human p97 more potent than the other. So it's relatively selective. And we also have mass spec evidence to show it really modified the cysteine we designed to modify. So with that data, got very, very excited. So we try to contact multiple people, multiple labs that we think we might be able to find more inhibitors. And luckily, there's Nathaniel Gray. At that time, he actually started his lab at Harvard. He connected us with a Scripps high throughput screening group. And that's how we start the high throughput screening. And at that time, the center is called MLPCN. And luckily, you only need R3 to start a high throughput screening. So all of this work has been published and summarized. Just as I put a timeline there for each publication, just to show you, drug discovery is hard, but if everybody work together, it's possible to move things to clinical and even to the patient soon. So you can see after the high throughput screening, we have feedback, which is the compound Donna mentioned also, and the potency in the bottom. You can see to do a process called SAR, meaning you make different change on the compound. And this is a great collaboration with Karen and Frank at Kansas University. And I have to say, I really appreciate my NIH program officer because the previous collaboration dissolved and they helped us to find Kansas University. Without their help, without their support, we would not have ML compound come out after this collaboration. And then Ray started a company called Clip Bioscience, and this the company quickly make a lot more compounds, and the phase 1 clinical trial started 2015. But unfortunately, there is some off-target effect, so the trial got stopped by FDA. Luckily, Clip also quickly figured out what is the off-target effect on the caused by CB-5083. So last year, Clip and NCI started this clinical trial with the new compound. So far, works great. So really looking forward to see the trial result soon. And interestingly, during this year, since I know Nathan, 2019, he is on top of literature. So there's this reference in the bottom showing just published, I think a couple of weeks ago in Brain Communication, ML240, which is the compound I put in the purple box, actually might have some effect to rescue the mislocalization of TDP43 and FAST in the VCP mutant motor neuron. So now one important asset which I spent a lot of time to help us identify defect is the compound to go after to do SAR is this dual reporter assay. And this is basically combine two different reporters from two different labs. So you can see the one on the top, this UBGAP, is a ubiquitin fusion degradation reporter. And the degradation of this protein depends on p97. The one in the bottom is OCC, the oxygen-dependent degradation domain of HIF1 alpha fused to a luciferase. And degradation of this one does not depend on p97. What does this mean? This means when we make this cell, and when you have any inhibitors or any gene, you want to know if this gene or inhibitor involved in p97 dependent degradation, you basically treat the cell with the inhibitor. If it only blocks the top one, the green one, not the bottom one, then we can claim it's relative p97 selective in the cell, not with purified protein. So this is a one way to help us filter out what might not be a selective inhibitor early on. So with the ML240 project and with some of the earlier compound we have, and luckily 2011, we got to join the p97 next program. And this next program was really great because I still highly appreciate. With our next program, I think the CB compound series will not also be able to move forward clearly. So we are the first project got to join, and the reason we got to join is University of Minnesota Drug Discovery Center, where I get my PhD. The director recommend us to apply to the program. And then since then, we have so many different inhibitors available, right? And the one now advanced in clinical is still the ATP competitive inhibitor. So my lab spent a lot of time trying to figure out or characterize those inhibitors more. And one type of the characterization is, well, all of the targeted therapy will have resistance occurred. And in Cliff's paper, they also show those mutations that occurred when they do RNA sequencing. So we characterize, you can see this pocket is where the mutation occurred when we treat the cancer with the compound. And the cancer cell is smart. They just, make the mutation on the protein and make the protein no longer being inhibited by the inhibitor. Then we want to figure out why there are these 2 different or 3 different regions. And we purify those mutant protein. So you can see the figure on the right is a steady state kinetic analysis. And the x-axis is just different ATP concentration. Y is the ATPase activity. So for wild type, the KK is around 5. And this particular mutant around the linker region, E47K, make the protein much more active. But the other mutation on N660K make the protein bind to ATP much tighter. So since it's ATP competitive, so if the mutant bind to ATP tighter, means the inhibitor also need more inhibitor to be able to inhibit. That's 2 different resistance mechanisms for the same drug. And now, since we make those resistant protein and also the cell line, so we want to test if allostatic inhibitor by the mechanism of action, since they bind different pocket of p97, ideally you should be able to block the resistant cell from growth, the CB-5083 resistant cell. So indeed, those 2 compounds, the compound Donna mentioned, the phenolindole and triazole, we can show it can block the CB-5083 resistance. Then I always got the question to say, oh, which inhibitor should I use to do the experiment, right? And so then I feel it's our duty to try to characterize those inhibitors more. And we have like different reporter cell line, different assay, but since my lab moved back to Caltech, we have access to this proteomics facility. So we start to use so-called unbiased approach. So the talk from yesterday, you can use proteomics to find the biomarker. Now, we want to use proteomics. In addition to find biomarker, we want to find how those inhibitors might behave differently in the cell in an unbiased way. And how do you do in an unbiased way? We just look at as many proteins as possible. Not all, because proteomics is still not as good as genomic sequencing. You can almost go to find every single gene. But proteomics is not that level yet. But for the past 10 years, it's increased from, you can find 100 protein. Now, easily you can find thousands of protein. So the mechanical plot here, you are looking at on the left figure is when we knock down p97 in the HCT116, it's the colon cell, colon cancer cell. Oh, I forgot to mention. And in addition to find how the different inhibitor is doing, we also want to find why p97 inhibitor might be potentially effective for the solid tumor, but not the proteasome inhibitor. So this plot shows when you knock down p97 by shRNA, so the one we highlight, shRNA level did decrease. The green dot is the protein level decrease that's more than twofold. And the red dot is the protein level increase more than twofold. And we validated the CHOP and ATF3. These are the ER stress marker we already use to do Western. So it's good to see they show up also in the mass spec-based assay. And this is the 3 arm of ER stress protein that we can identify and also being dysregulated or the differential expressed when we knock down p97. Then we want to do similar experiment for the inhibitor. So you know the difference between knockdown and the inhibitor is knockdown you need to reduce p97 protein level for over 3 days to see the effect because this process genetic method is just slow. For inhibitor, you can treat at different time point and see what happened at different time point. And that's what we did. Say if the blue bar is 2 hours and 6, 10, 18, 24, we do up to 24. And what Feng and San did was basically overlay the differential expressed protein of at different time point with p97 knockdown data, the data I just showed you before, and compare these 3 inhibitors. So you can see CB and NMS has more overlap and it's time dependent when the time point at 24 hours is more overlap with the knockdown. And this is another view of that data with a selected protein. So those are different protein, and this is the heat map. On the right, last row is p97 shRNA knockdown, and different compound treatment at different time point. So you can see CB and NMS looks relatively similar for some region. There's still some difference compared to the knockdown, but NMS, it looks quite different in this assay. So then all of the data I show you, we use the method called label-free quant, meaning you don't label anything, you just analyze all of the sample and the mass spec tell you what they can detect. So, and the method is good because we have like 50 samples to compare. And yesterday you heard about a method called TMT labeling. So for there are plus and minus for both methods. The one for label free, you can do as many N as possible. For TMT labeling, we try to compare the data in one set, and so far we can do 16. So a Thermo Fisher only show you 16 plex, meaning you only can compare 16 samples at one time. So because of that, we focus on 2 compounds, CB and NMS and MG132, and only compare 6 hours and 24 hours time point. And this PCA plot shows you the region 6 hours separate, or the experiment we done at 6 hours separate well to the 24-hour time point. And this is not surprising. The one I just showed you, the heat map change also when we treat with compound more, there are more things might change. But the earlier time point might tells you what are the protein marker can be affected by p97 inhibition earlier. So earlier seems to be more potentially direct target. And the later time point could potentially come from a secondary effect of the direct target. And this is a pathway analysis of the protein specifically downregulated by p97 inhibitors. And some of them are not surprising, like cell cycle, because p97 is known in East Coast CDC48, and that's what it first identified in regulating cell cycle division. So the cell cycle protein is one of the one specifically changed by p97. And these are the pathway specifically upregulate. And again, we already know p97 is essential for ER stress, ERA pathway, ER stress, UPR. So the protein specific upregulated by p97 also falls to those pathways. Then one of the key we want to know is what is different between inhibiting proteasome and p97 in the cell. So this is one of the protein called cyclin B1. And you can see at different time point for MG132 treatment, you see the protein level keep increasing and increasing up to more than fourfold at 24-hour time point. But for the two p97 inhibitors, CB and NMS, you can see the protein level actually decrease. And this is not just for one protein. We observe several cell cycle oncoprotein actually specific downregulated by p97 inhibitors. So now we want to figure out if those downregulate effect compared to MG132 is the reason MG132 is not effective in solid tumor. And that remains to be determined. So when we are working with Donna and NCI team on the NMS triazole analog project, one of the observations we keep observing, even like early on when we just use NMS as a control, when we're doing other experiments, you can look at the picture on the A picture. The circle, these are basically a 6-well play image of 6-hour treatment and 24-hour treatment. And the top is DMSO, which is the vehicle. And the bottom is NMS compound. So you can see, 6 hours, the media color change. 24 hours is very obvious. And what does this mean? Means the pH of the media change and likely become acidic. So we did measure the lactate concentration, and they did increase. So, and typically this might be a function of changing in mitochondrial function. So Feng and San also did a C-host assay. So you can see at the for the 6-hour treatment, 2 micromolar NMS compound inhibit the oxygen consumption very significantly. So we want to figure out, and we want to figure out if this effect, what pathway might be this effect by NMS. And you can see the overlay of the Venn diagram of protein, they are differentially changed by 3 inhibitors was compared. And there are 219 proteins that are more specific for NMS. And when we do pathway analysis of these 219 proteins, you can see those are the pathway related to mitochondria. And the lactate formation is also specific for this compound because the CB or UPCTC MG132 doesn't have the same effect. So we think somehow NMS might be affecting this glycolysis pathway. So we use another pharmacological inhibitor called 2DG, which is a glucose analog. And with the combination of NMS compound and 2DG, we see the in the presence of 2DG up to 4.8 millimolar, we can rescue the effect of NMS A73 in the lactate formation. So in the media change color become acidic. And on this C panel shows you when we combine both drugs, we can see a synergistic effect. And this effect, again, if you look at the D panel, it's only shows up for NMS compound, not the other compounds. Then to figure out if this effect we are seeing is due to inhibiting p97 or not, Feng and San make the NMS A73 resistant HCT cancer cell line. So you can see the A panel is basically compare the cell cytotoxicity, the cell title growth assay for NMS compound with compared to the parental wild type 9 and 2 NMS resistant 9. So you can see there are 2 9s and the second 9 is more than 10-fold resistant. And the interesting thing is we cannot keep increasing the concentration to make it 100-fold, but we can do that for CB compound. So CB compound, we can make resistant 9 for more than around 100-fold resistant. But for NMS compound, 10-fold is the ceiling. We cannot make more. And at the time, we don't know why, but I think now we know. And so these two resistant cell line is a good tool, but we first need to validate the resistant cell line still can function as it's supposed to be. So we use different inhibitors to verify that. And then we compare, so the panel C is a pathway analysis, compare the parent cell line treated with the NMS S73. And the second one is the resistant cell line treated with NMS S73. And you can see what tells you is what are the pathway, the pathway that disappeared tells you those are the p97 dependent pathway, because those are the pathway render when the cell line is resistant to this compound, and due to mutation on p97, we also verify that. But there are certain pathways still shows up. So the top panel, and on the right hand side is the resistant cell still will produce lactate. So in this resistant cell line, you use different concentration. You can see the lactate still produced. So from this experiment, we confirm, or we conclude the lactate formation is a p97 independent event. Then how do I find what is the cellular target for this event? So we utilize an assay that look at protein stability in change, thermal stability induced by binding to compounds or binding to the protein. So you can see this figure, the melting temperature of a protein is a constant, Tm, at a certain condition. So if you if the compound bind to the protein, and this binding make the protein more stable or less stable at different temperature, then you will see this Tm shift. So this is a very fundamental phenomena for the protein biochemistry.
Tsui-Fen, I want to make sure we just time check. I mean, we've got a couple more minutes, but
okay.
I want to leave some time for Q&A.
Okay, sounds good. Thank you. So then basically, we use the same assay you doing by mass spec to find the off-target effect for p97. I'll just get this for NMS compound. So this is a thermal proteome profiling with the compound and compare with DMS. So you can see on the panel F, those are the protein we can identify has thermal stability shift in the presence of the NMS compound. And with the homology modeling and with overexpression, the protein in the mitochondria complex 1. And also at the same time, there's a paper published actually show NMS is a dual inhibitor for mitochondria oxidative phosphorylation. So we think this is the potential off target of NMS compound on the mitochondria complex 1 to cause the effect. But this might still be a good thing, because when you have a compound can block both pathway for the cancer therapy, we can have synergistic effect for the treatment. And the future goal is, we want to work with, whoever is interested, try to develop a p97 complex specific inhibitor or activator. And we already performed a high throughput screening with mutant p97 bind to p47 cofactor and trying to find a compound, and this is ongoing. And finally, the p97 is so complex and different cofactors. So I think both good chemical tool and genetic tool will be important to help us to understand. And all of the assay we done in the cancer cell can be done in the VCP disease cell. And for my postdoc, we'll discuss some of the data we already have on that part. But, we need, people to work together as a team and to tackle those questions systematically. I'm not going to talk about it. So basically, these are the people who, bravely to work with me, and I really appreciate their hard working and their idea and their contribution to all of the project in the lab, and my collaborator here, and funding.
All right. Thank you so much for that very informative presentation, both are very informative presentations. Do we have questions? P? Are y'all aware?
Yeah. There's some reports, but you don't know whether they're old programs, right? And so other than Cleve, I don't know of any active programs from companies, from big pharma, I would say from big pharma, right? Most of the work in the literature is from academic groups. I think there was one from Merck KG, but again, you don't know whether it's an active program or a finished program. So I don't know if Tsui-Fen, if you have any more information.
Yeah, company I know of, yeah. And in terms of the question about inhibitor activator, I think we can hear the extra presentation, and then we're happy to discuss more from there. Yeah.
Yes, that's a great segue into our next session, which we're going to hear from some of the poster presenters, because there were diverging opinions or diverging posters, which was wonderful to see. Okay, we do have Hemo here. How do you envision cofactor specific inhibitors if VCP always uses its D2 ATPase activity?
You want to take that one, or done, or you want?
Well, I mean, so one way you could think about it is binding to that interface, right? And so they would be very different types of inhibitors. They would be inhibiting the VCP cofactor binding site, right? But I think there's already data in the literature that says even the active site inhibitors, the allosteric inhibitors, have different effects on p97 cofactors, right? So there, I don't think we understand why it does, but it seems like it's feasible. You see Tsui-Fen's published papers where you see very different structure activity relationships in when you have p97, p47 than you do in other systems. So I think it's feasible. How to do it, I think we have to still learn how to do that.
Yeah, and I do think it's not an easy task, but you need both a good chemical tool and good assays. So basically, once you have the inhibitor, we can easily purify protein and mix the cofactor. But what is the cellular pathway? It's going to report that particular cofactor activity, and that's the important one. And we know each cofactor complex is different. It's a different conformation, and the substrate. We also have another mixing information we don't know about the substrate. So it's possible. It requires a lot more work. It would be.
No, I think Michelle has an important comment. You know, the allosteric inhibitors offer an opportunity, maybe more of an opportunity than the active site inhibitors, because you think that site might be more flexible and more reflective of other conformational changes than the ATP site, which is pretty conserved.
All right, well, let's go ahead, and we're going to stay in this session. Nobody leave. Chang, do you want to come on? But Tsui-Fen and Donna, thank you again for your presentations, and I know you'll be chiming in some more on this. So